nucleic acids, where we are clearing some of your misconceptions as far as nucleic acids are concerned. We've grown up hearing DNA, DNA. Smile, do we know what DNA is? Yes, and I hope from the previous videos, we now know what DNA is, where we were able to talk about the structure of DNA and how the DNA can be copied to get another DNA that is exactly the same as what we have. So previously we talked about DNA replication, the enzymes involved in DNA replication, both the leading and the lagging strand, up to where we stopped. Now today we are going to proceed with mechanism of DNA replication. Now there are theories that try to explain, how DNA is going to be produced after copying the DNA and you formed your strands, the two strands of DNA from the parent strand or from the parent strands. So these theories try to explain what happens after the two strands have been formed. And there are three, whereby in one, it believes in conserving the parent DNA and the new DNA synthesized. So this is the conservative hypothesis, conservative hypothesis. Here, this one believes or tries to hypothesize that the double helix or the double helices, both the new and the old, they remain intact. That the old strand of DNA remains intact. Okay? And the new strand, the two new strands of DNA formed, they also form their own DNA. That there won't be any mixing of the two strands of DNA. So this one, after synthesizing the entire strand of DNA, then the original DNA, it remains alone, and the new double helix that has been formed, it also forms its own DNA. So that is the meaning of conservative. You try to conserve, our nucleic acids. And there are tests, the experiments that have been conducted. So with conservative, you can have a look. This is our DNA, or our parent DNA, that is going to be coopted to unwind and then the enzymes, the entire process of DNA replication. Now the new strand of DNA, both the complementary and the template strands that will be formed, they will diffuse out of this old DNA and form their own DNA molecules. But the old strand, it also remains intact, and the bases will form hydrogen bonds again and form the old strand of DNA that remains intact. That if further replication occurs, maybe if you replicate, you also form another intact strand. Maybe this one is also intact. This one now becomes the parent strand. And you also form a new strand of DNA. The same applies if you continue and copy this. The same will happen. You form the parent strand of DNA that is intact and a new strand of DNA. So that is the conservative hypothesis. hypothesis. It conserves. Okay? It conserves. So result is that in each generation, one of the DNA molecules is exactly the same as the original molecule. Then another hypothesis is the semi-conservative. Semi means half. Okay? Conservative means conserve. Hypothesis comes from conserve. Okay? Semi-conservative hypothesis. Now in this hypothesis tries to explain that both the original strand of DNA and the parent strand of DNA, they can stay together, and you form the two DNA molecules with both an old and a new strand. So after you've unwound the double helix and you've copied the double helix, each old strand serves as a template for the formation of new strand. We have appreciated that as far as DNA replication is concerned. And you form a new strand of DNA. Now, after forming that new strand of DNA, it won't diffuse or it won't come out of the DNA. No. The new strands of DNA which have been formed are retained within, and they can bind with the old template strands of DNA. And the newly formed DNA is going to be half the original strand and half the new strand that has been formed. As you can see from this drawing. So if this is our old DNA, yes, it will unwind, and after unwinding, it will expose the organic bases and then DNA polymerase to come and synthesize new strands of DNA. And you form new DNA strands. Now, this one, as you are forming the new strands, okay? So as the enzyme, this is your replication fork. So as you are forming new strands of DNA, the new strands can bind with the old strands. And then you form that DNA structure. So that the new DNA molecules formed, they will contain half the original template strand, which is the blue one, as you can see, and half the new strand of DNA, which is the red one. Now, how is this achievable? How is it possible? You know, the law of complementarity. The binding is complement. They complement each other. So as a result, this new strand that is formed, like I told you, it is going to be exactly the same as this old strand. And this new strand of DNA that is formed here, it is going to be exactly the same as this old strand here. So there is no reason why the two cannot bind. Why the two cannot bind to form one DNA molecule. So that is the conservative hypothesis. Now with dispersive, this one is confusing.
Excuse me.
Yes,
So in this semi-conservative, one helix is of the old and the other one is the new.
exactly.
Give
Exactly
me
that.
the slide we are on for like 2 seconds. Thank you.
Okay, you're welcome. So exactly. In semi, it's okay, you can mute. In semi-conservative, it's half. Okay? Half conserve But in conservative, it's fully, the whole of it, the new. So the old DNA is conserved. Okay? But in semi, there is half conservation. In that half the strand of DNA is going to be from old template strand, and half is going to be the new complementary strand that has been formed. So that's your replication. Now with dispersive, this one is just confusing. This one is simply confusing. Now in that, the new synthesis, the synthesis of DNA will be a mixture of both the parent and the newly formed strand. This is just confusing. Yes, we have seen that in conservative, you would have the old aron and the new aron. No tampering. In semi, you have half strand old, half strand new. But now here, in both the two DNA molecules that are formed, you will just have a mixture, okay, of old. Maybe one backbone. This one is going to be the new strand of DNA, and then the other one, or the other backbone, is going to be for the old strand of DNA. This is now old strand of DNA. And then the next one becomes the new strand. And then the next one becomes the old strand. So that's why it is dispersive. Dispersive. You are dispersing between the old and the new. The old and the new. The old and the new strand. So it's just confusion. I call this confusion. Okay? So you have your DNA that is formed. You have segments of the new strand and segments of the old strand. So you can have a look at this. Just have a glimpse here. So if this is our old strand, our old DNA, the newly synthesized DNA molecules, you can see they are made up of old strands. The one which is blue, those are old strands of DNA. And those segments that are faint, those are newly synthesized strands. And you can have a glimpse here. This is a newly synthesized strand, newly synthesized strand. Okay? And then this is the old strand of DNA. So this is really dispersive. Okay? This is dispersive. So you continue from generation to generation to generation to generation. Okay? Yes. So those are the three theories that try to explain the mechanism of DNA replication. How actually our DNA is going to be formed. So the DNA involved, the here replication involves fragmentation of the parent double helix, and then after fragmentation, then you intermix the newly formed strands and the old strands of template DNA. So it's as simple as that, guys. Yes, so that's basically the theories, the mechanisms that try to explain how DNA can be copied. And it can, by the way, this one can be experimented. It can be experimented. Whereby, however, it is going to be kinda technical, but if you get your bacterial cells, your bacteria, that are replicating. Okay, and then you supply them, you first give them, you put them in culture solutions, okay, containing nitrogen has isotopes. We have nitrogen 14 and nitrogen, is it 16? I think nitrogen 14 and 16. You can fact check this. Now if you first put them in 14, they will, in culture containing organic bases made up of nitrogen 14, they are going to replicate and form the DNA. Now after they have replicated, when you transfer these bacteria and you put them in a culture solution containing this other isotope, the organic bases with isotopes carbon 16, they will also replicate their DNA. Okay, they will replicate their DNA. Now there will be first replication and second replication. Now carbon 14, in the first they used 14, in the second they used 16. Now to test for this hypothesis, we shall try to separate this DNA. 14 and 16, they have different weights. One is 16, another one is 14. These are atomic masses. So if you use fractional, we shall use centrifugation. Remember we studied centrifugation. Centrifugation, a centrifuge. We shall be able to test for this hypothesis, for these three hypotheses, whereby if you form two DNAs, one is going to be formed up and you form another one down, then it means these are totally different strands, different DNA molecules. Okay, and this one will support the conservative hypothesis that the new DNA, which is heavy, okay, which is formed from a heavy, which, remember, we gave this carbon 16, so it has been formed from organic bases made up of carbon 16, so it will be heavy, so it will be the one set at the bottom. And then the other one, which was light, the old one, which was formed from carbon 14, it will be the one to settle on top. But if you fail to form the two separate mixtures, you do centrifugation and you have one complete one DNA. There is nothing like separating between the two DNA molecules. Then that one implies that both the old DNA and the new DNA synthesized, they have been mixed. Both carbon 14 and both nitrogen 14 and nitrogen 16 have been mixed. Okay, and so they cannot separate. And so that one confirms, that one supports the dispersive and the semi-conservative. So that it is really a clear indication. Okay, it is really a good illustration. It's really a good experiment that tries to support these theories of DNA replication.